Okay, so again, this section is going through just the introduction to cells, how they were first discovered, and just kind of a brief overview. So, we are all made of cells. As our warm-up talked about, they're the basic unit of structure and function in living things. So they are what we are made up of. They help us perform tasks. They keep like our organs functioning, all of that. Okay. So cells form all of the parts of an organism, and they carry out all of an organism's processes or functions. So how things are regulated, how our heart beats, how it moves blood, how we think and process information. It's all controlled and functioning due to cells. Okay? So think about if we described how our pencil worked. We would explain the components of the pencil, maybe what each part of the pencil did and how it functioned. So the eraser is able to erase and remove those marks we make. There's a spring in there that helps move the lead through the pencil if you're using a mechanical pencil. There is the lead in there so that you can write something, things like that. So with the structure of living things, that's determined by the variety of ways in which cells are put together. So some of our cells are arranged to be an organ or to help digest food. With a tree, cells are arranged to form the trunk and then the leaves and the branches. And plant cells especially are set up in specific patterns to create that leaf and that tree. Yeah. So then an organism's functions are the processes that enable the organism to stay alive and reproduce. So as long as we're able to continue functioning, our body is able to continue to move and process different things. We are able to stay alive. Some of these functions include obtaining oxygen so that we can breathe, getting rid of waste, whether it's our body breaking that waste down or how it removes it, obtaining food, and even growing is a process to help stay alive. And then cells are involved in all of these functions. And they have specific tasks and functions that the cells kind of control or process. Okay, so cells are very, very small. I'm sure, hopefully you guys know that. But if you thought about the space on your hand, or if you looked at your hand, 1 square centimeter of your skin surface contains more than 100,000 cells. Okay, just that little itty bitty centimeter. So if you put that all together and measured how many centimeters your skin alone covers, that's a lot of cells. And then think about your organs and your blood and the neurons in your brain and everything else that you're made up of. Those are all made of cells as well. Okay, so no matter how closely you look at your skin or the surface of something with just your eyes, you're not going to be able to see those individual cells because they're so extremely small. Just like with every other cell. So until the late 1600s, no one knew that cells actually existed and that we were made up of those specific individual cells because they didn't have a way to look at it and actually see that there were different objects creating a bigger object, which was us. Okay, so around 1590, the allowed people to look at very small objects. It was like a magnifying glass, but even bigger. That it was able to take a small object, make it bigger. You could see more details and different things like that as well. And through this, the invention of the microscope made it possible for people to discover and learn about cells. They could finally see that a large object was actually made up of those smaller cells and individual pieces. Okay, so overview of a microscope. We will get into this more at the end of section 2.3. But a microscope is an instrument that makes small objects look larger. Some microscopes do this by using lenses to focus the light, so then it's like more concentrated and like brighter, that you can look at a more specific smaller object. The lenses used in light microscopes are similar to clear curved pieces of glass or plastic, like eyeglasses. So if you have a pair of reading glasses or glasses, the lens is similar to that. It helps magnify the object a little bit more. There are different types of microscopes as well. So a simple microscope contains only one lens. And then a light microscope that has more than one lens to kind of like layer it and help with that magnification is called a compound microscope. Okay, so you're putting multiple lenses together. There's even other types of microscopes as well that again we'll get into. And when we do our labs on microscopes, we will use and look at compound light microscopes. One of the first people to observe cells and how he noticed and determined what they were was English scientist Robert Hooke. And he actually built his own compound microscope, which is what's in the picture. And during his time, that type of microscope was one of the best out there. And had either the most clear view or just the setup and function of it worked the best. So in 1663, Hooke used his microscope to observe the structure of a thin slice of cork. And one of the things with microscopes, when you do use them, you have to have a very, very thin piece of something so that the light can pass through and you can see all of those components and objects that you're looking at. But his microscope obviously had an eyepiece. It had that barrel or tube. There were some knobs to kind of help focus. He had lenses in it. There was a specimen holder. And then he also had that set up for the oil lamp to produce the light. And then that water flask helped expand the light and make it a little bit brighter and more focused on that specimen. Okay, so with those cork cells that he was observing, cork is made up of cells that are no longer alive. So cork is something that was once living, but once you have that cork, it's not. And so to Hooke, when he observed that, it looked like the cork had tiny rectangular rooms or structures. Okay, so he called those empty spaces cells, which is a word meaning small rooms. And you can think if, like, this isn't a fun picture to think of, but if you think of like a prison cell, or if you think of like monks in monasteries and they have their like cells, they're just kind of like small rectangular rooms and enclosures. They all look the same. They're kind of right next to each other, all lined up, and they have just that empty little space. Okay? Okay? So the next person to look at cells around the same time of Robert Hooke was Anton van Leeuwenhoek, and he made observations of tiny objects with microscopes as well. So this was his microscope set up in the picture. It's obviously a little bit different, but it has kind of that flat piece, and then there's a more focused lens that is kind of like a magnifying glass almost, and it helps magnify that object. So he just built a simple microscope. It only had that one lens, whereas Hooke's microscope was a compound microscope that had multiple lenses. Now, Leeuwenhoek looked at drops of lake water. He took scrapings from like people's teeth and gums, and even looked at water from rain gutters. Okay, so you can think like back in those 1700s, like people obviously didn't have very good dental hygiene. So there was a lot of bacteria and like gross stuff in your mouth. So taking that scrapings from like people's teeth and gums, he was able to look at that bacteria and those different particles and pieces as well. So in many of these materials, he was actually surprised to find one cell or single-celled unicellular organisms. And one of the cool things was he noticed that many of these organisms moved. Okay, so some whirled and like twisted and twirled around. Some of them looked like they hopped around, and then even some like shot through the water like fish. And so what he did was he called these moving organisms animalcules, something like that. Essentially meaning little animals. So that's what he kind of thought of them as. They looked similar to like some sort of animal or organism that moved and had similar characteristics of an animal.